starting this week. Okay, so usually how it works is like that. I will leave it in the class totally dismantled. Then in your pad, you can grab it. Then you set up your camera, and then you time lapse yourself. Right, the fastest group wins. So, what do you say? You want to take a photo of this? Okay, so I'm going to leave this in class for today for you to analyze. Right? This is the final model you should build. Oh, yeah. Can, of course I can. But something will happen if I copy, right? Okay, so this particular model, you have 1 hour to do it.
How long did it take you?
To me, like 15 minutes. 15 minutes, just kidding. No, it took me really long also. So, but I timed myself. I could do it around 50 minutes. Okay, I could build it in 50 minutes. Okay, so I'm one person. Okay, but then this is because I study biology, I teach you all biology, so I have an advantage. Okay, you can beat my timing, then you very good. Okay, so in 1 hour, this is doable. I said this timing because it's based on past year's challenge. I see how long roughly a pair takes, around 50 minutes. Okay, so you can have a look. This is a model of the DNA. Right. Today, we're going to continue on. The knowledge you gain here will give you quite a deep understanding of how you're going to build that. Right? Who's here? Who is this? Zoe. Who? Enough data? This one. Okay, so can I get you to stand by this particular CA? We're going to continue on from the previous lesson. Okay, today we're going to learn how to build the DNA by 2D first. Okay? Understanding how we build it in 2D will give you a better idea how to construct it in 3D. Right? I'll give you some tips and tricks on how to identify the atoms and the model once you actually start using the real deal. Okay? In groups of, around 10 here. Okay, I'll just distribute first. I'd like you to work in groups of, okay, turn around, work in groups of 4. Okay, so just 1 pack, okay. 2 pack. Yes, thank you. Okay, yeah. Pricing how? Oh, pricing what? Yeah. Okay. Then your passage, your cost is pricing for us. Have you, Jaden, has the recording started? Okay, alright, okay, I'm going to do a quick recap of what we did last week so that we are all on the same page. Okay, some of you weren't in class. Okay, so today, first and foremost, we recap what a nucleotide is. Secondly, we're going to perform condensation reaction. Last but not least, we are going to look at hydrogen bonding between bases. Okay, 3 things you will do today. Number 1, let's recap the structure of a nucleotide. What are the 3 distinct parts of a nucleotide? What's at the core? Okay, there is one pentagon thingy. What pentose? Pentose. Okay. Pentose sugar. Yeah. Okay, it's a sugar. And then there are 2 kinds of pentose sugar, right? One is a ribose. One is a deoxyribose. Okay, so that's the core of it all. In the model over here, you will actually find all of the sugars at the sides. Sometimes we refer to this as a sugar phosphate backbone. You look at this model over here. Sooner or later I'm going to drop this on the floor. Okay, can you see that I have a long chain of black at the side? Yeah, that's where all the sugars are. The sugar actually is not in the middle of the DNA, it's at the side. One chain, if I turn it around, another chain going down the other way. Yeah, so we call it the sugar phosphate backbone. Okay, so that's the first thing. Now I want to teach you a tip. Each carbon is actually numbered. Yeah, this is carbon number 1, carbon number 2, 3, 4. And then what you'll find, right, is that there's one oxygen coming out, and then you'll find the next carbon number 6 over here. Carbon number 5. Okay, so that's what you want to look out for. And carbon number 1 is where you bond to a base, right? Okay, where do you find all the base? The base is the middle portion of the DNA. All the bases are going to be concentrated in the middle, right? Later on you will see it for yourself. How many kinds of bases we have?
3, 5.
Okay. 5 bases, but can categorize into 2 categories, right? One category is called purines versus pyrimidines. If I just use the alphabets to represent, what's under purines? A and G. And then my student taught me that pyramids are very sharp, therefore they can cut. Okay? Yeah? So last but not least, at carbon number 5 is where you will find a phosphate group. Yeah? And this is what a nucleotide is. This is what is repeating along the entire DNA. We just use the same thing and we connect them together to form 2 chains of DNA strands that we will link together. At carbon number 2, we have some variations. It could be a OH or it could just be a hydrogen bonded there. Last but not least, at carbon number 3, you will always find a hydroxyl group. Okay? Right. So now, here's tip number 1. In order to construct this DNA, you must have a very keen awareness of the numbering of the carbons. If you know which carbon is which, it makes it easier for you to connect them together later on. The moment you lose track of which carbon is which, it's very hard to piece them together. For our first activity today, we're going to try and error. I'm not going to tell you how each nucleotide links together. Okay, I'd like you to try it for yourself. I'd like you to take out from the plastic, you'll find small little nucleotides. So continue from last week. Yeah? Can you find the nucleotides inside the folder? Not the big ones, the really tiny ones.
Wait, then sorry, we have 2 packets, you only need 1, right?
One packet will do.
Okay, let me just...
Okay, can you find the nucleotides?
Just stop that. Yeah.
Okay, here's the first puzzle for today. How can you perform condensation reactions such that you can link up all the nucleotides together in a nice repeating pattern? Okay, that's the first puzzle for today. Without me teaching you, based on your current knowledge of how condensation reaction works, they need to extract out water. How would you link up all of the nucleotides together in a repeating pattern?
It's the ribose.
Okay, ready? Your group has
And this is the ribose. Because like
7 minutes to figure out this puzzle.
This OH to this
when you always do this.
Like, need to
Like,
connect.
you know, there's one here, there's a ribose or deoxyribose.
This is deoxy.
Yeah, this is deoxy because it's only a H. Because it's only a hydrogen, they won't
How do you know
write
it's
it down.
deoxy or this?
Because if it's a ribose, they will write OH. But because deoxy only hydrogen, they don't, like in carbon skeleton, they just write, you don't put the H.
Oh,
Yeah. So
okay, but
it's
all
all
of these are
deoxy.
deoxy.
Yeah.
Okay, then how do you bond it?
How do you bond it? Condensation reaction.
We don't check, we just try.
Condensation, C and O will connect to this part, then condensation reaction probably like H2O from here, then you connect to like, then they just. Why did he ask to connect?
this DNA one.
This is the base. The base. Like what base it is.
Like this, then
Wait guys, 1, 3 is the base, right? But
Yeah,
then
all
why
these
is it
things
numbered?
are. I don't know why it's numbered.
it's not one.
This is a 1, this is a 3. This also got 3. I don't know why the bases are numbered. I'm not sure yet.
Yeah.
Why are the bases numbered?
Why are the bases numbered? I numbered it, or rather we numbered it because we don't want you to focus on the bases yet. Okay, the bases are actually A, T, C, G. All the number corresponds to one of the base, right? But we want you to focus instead of how it comes together rather than the bases for now.
Yeah, I think I know.
Yeah, sorry.
Yeah, I think. Different.
Okay, so ignore the numbers for now. Okay, ignore the numbers.
It's either, I think it's either.
But then
They don't join to the base, they join to the phosphate group.
you need this and do this.
Because remember the saying, then phosphate group combines with another phosphate group. The base is just a base. Correct?
Oh wait.
Wait, is it connected like on top of each other or as a line?
I don't know.
Where can you extract out water from to form bonds?
Do they connect to the base or do they connect to the phosphate group?
Okay, so that is the challenge here. How will you connect them together such that you can keep connecting more and more and more?
Should be the phosphate, right? Because phosphate got. Wait, no,
Wait,
cannot
then
be
can
the
we
phosphate.
connect
because
as
one-
like that or like that?
That's the thing, it's up to you. So you figure out this puzzle. How will you connect such that you can keep connecting and it doesn't end in a dead end?
What will we connect it to?
And that is the thing for you to figure out. How do you connect such that you don't create a dead end for yourself?
So
And you
whenever
can
you
keep
connect two,
connecting.
must have a O and it must have a H.
I
see.
feel like we should.
Must have O and must have a H.
Right, okay, right? Because you need to extract water out, right?
Yeah.
Yeah, I feel like it's like that.
I think it's keep connecting this part because this
So
part
it's like-
is
goes like
moving.
that.
Yeah, it goes this way. 'Cause
So if the only H is here, you need to take out H2. Then this one can connect to the base, right?
can connect to base, but I thought I think also can connect to phosphate.
How will you connect without reaching
Oh,
a
but
dead
he said
end?
must connect until it doesn't reach a dead end.
Connect
So
without
whenever
reaching
you
a
Connect
connect,
like that, then cannot do. Done.
no, as in you just keep connecting phosphate group. Don't connect this one.
But you're
So
supposed to, having like nucleotides together. Yeah, not just phosphate.
here no H, but if here no H,
Yeah, so
oh got
but
H,
yeah, you can do like that.
then can like that can.
So you
you
take
connect
out
like
OH
this,
from here.
and then this one go to that side.
Wait, you take out OH from here, you take out H from here.
No, you can take out H, O, and then one H from here.
Yeah. So if yeah, okay.
Then this one
Then just
connect
keep
with
on
this
going
one.
on.
Then like that unlimited already. H2O.
Then it's kind of like in a staircase position.
Wait, do you connect it to this NH or do you connect to this? Here got NH2, this one NH. So you want oh, some only have N, this one is not really.
Oh, it's different bases, huh? Oh, so these two are the same.
Yeah, does it matter whether we connect to NH2 or NH?
I don't think it matters.
Okay, then we just keep connecting. So like that will be unlimited, right? Because every base will have at least one H and then phosphate group will have H. Then it will be unlimited. Lor.
But you can only go in straight line.
Yeah.
Cannot like
It's just like that. Then it won't even be like straight. Oh, that's why it's this technical. Because it keeps like. So this one
it's a staircase
is.
for me.
Yeah. See, just now I see top.
I think it's one.
Yeah, like that. Like that.
Yeah, so it's staircase. That's why it's technical shape because it keeps turning. So that's why.
So you see the long way, and then
Like stop shaking at it.
Or you bond this one.
Wait.
Okay, so there is a way biology does it. What you've done,
Oh, no, it's looking for me.
okay, very possible. Now, if you like to, if you look at your notes, see how biology.
Oh yeah, so we don't arrange it like in a street, we arrange it like a subfield kind. But then like
Yeah,
becoming bigger.
circle staircase. Huh?
Now this one then.
Anything.
Oh, because it's 3D. So
But NH2O this.
yeah, so minus H2O.
You take out 2 of it. Then you take out O.
Then they will combine and to H.
Then add to P of.
Okay, if your group thinks you found a way to connect them together without dead end, let me know.
Oh, we did it, right?
Yeah, we did.
I don't know.
Okay. Oh, our group decided, wait. Separate them. Yeah. So we connect the phosphate group, right? See this? Connect to
The base.
The base.
Ah.
See here there's NH, then the NH2, then
Yeah.
here got HO. So we just connect it. Then NH2 and the HO. Then
I
we
see,
can
so
take away
the
a connect.
water comes out.
Yeah, then it's unlimited one. Because
Yeah.
then the base can connect to phosphate group.
Okay.
Then just keep connecting.
Yeah, fascinating,
We
yeah.
wanted to ask like,
Yeah.
okay, some of them, right, there is, wait, where's the third one? No, must be the one, yeah. two. So does it matter which one you want to, which one have candidacy? Does it matter?
I would say that in theory, this one might be easier to extract from because at the moment over here, okay, I think this one in chemistry, this one might be easier. Because this one over here, let me see, 1, 2, 3, 4, 6, 7. Do you intend to pull out 2 hydrogens from here or just one?
We could pull one, we could pull two also. Because we are
okay.
connecting it to this phosphate group, this OH.
Oh, so you're going to take the OH from here?
Yeah, we could pull out one, we could pull
Oh, then technically both can.
So both can.
Okay, yeah.
So which one will it bond?
Okay, so right, in theory now, you seem to have found a way to connect them all together, right? Without a dead end, right? Okay. Now you can open your notes, look at how the cell does it. Okay? How does a typical cell create DNA?
This one?
Oh, your notes. You look at your notes, and you see how the cell does it.
Oh yeah, same as us.
Is that the same?
Yeah.
Okay, around 1 minute more. So how does the cell do it? How is it different from yours?
Slightly different. They bond it to the bottom.
The 5 carbon phosphate at the end of the 3 carbon linkage of the acid. So 5 carbon of the phosphate.
Oh yeah, correct.
Okay?
All are 5 carbons.
Oh, I mean like 5 carbon. Yeah, okay. So
Yeah.
phosphate end of this to the 3 carbon end of the nucleotide.
Wait, what page are you on?
Since
Then we are not correct, but they bond to the bottom one.
yeah, so we're not correct.
So we're not, but ours is also correct, right? That
It's
in
born
theory
from
we are supposed to work.
it's born from here.
Yeah. So they bond in
Oh,
a
so that one is like that.
Wait, no, H2O, H2, then O. So they bond like that. Oh, in a way also staircase, see? Also staircase going.
Oh.
Yeah, that's why there's the clinical curve thing.
Oh, we came out with another.
Then why does it bond in this way and not our way?
Yeah, it's just for me, time is up.
I think there should be more than one way.
They do like that.
Right?
Oh yeah, it's like this now.
No. So, let me see. Where do you extract the water? So usually at the end of the you want, right? You see the biomolecules are all about patterns. So
Yeah, I see that.
let's see how biology does it.
8 below 5. Because that I'm the only one that has 0.
Give me the recap.
Look at how people cells are.
Whatever you can say.
Wait, then how much the 0 below?
You all do the same way as the cell?
Hey, Jaden, you want to get a... Oh, Jaden. 0 below. Just take it. I don't want to take it.
Okay, I'll take it.
Hey.
Oh, I want. I want this one though. I'll treat you. I'll treat you.
Okay.
I get it.
I've gone around to look at most groups, right? What I'm very happy with, number one, is that all of you managed to connect them such that you don't hit a dead end. If you notice in biology, at least for this topic of biomolecules, it's all about patterns. That we can create a repeating pattern so that we can elongate a chain. And you've successfully managed to do that. That's number one. Number two, you also found very good functional groups where you can extract water out from. That's how you will perform a condensation reaction. Although, now I'd like to draw your attention to how a typical cell does it. How does a cell link multiple nucleotides together? Did your group do it this way?
We did it after checking.
Once again, I want to say there's no right or wrong. Because in theory, if you did it in the lab by yourself, you could have connected them this way. However, under cell conditions, how do they really connect? Okay, if you go to your own notes, I believe you can access your own notes. Okay, you access your own notes and you scroll down to the second page. This is how, under cell conditions, nucleotides can join together repeatedly in a pattern without hitting a dead end. If I were to ask you now, which carbons are involved? Can you tell me? And which carbons do they join together?
Carbon 3.
Okay, carbon 3 seems to be one point where the OH is at. And it joins also to carbon 5 where the phosphate group is at. Right? So the pattern goes like that. At carbon 3 and carbon 5, where the phosphate group is, they will connect to each other and extract out a water molecule. And it repeats itself over and over and over again and form a really long chain. I saw some groups, very interestingly, they connected the base to an OH group, right, or phosphate group, right? Why can't that work in the biology context? You'll find later on that it's very important to have the base available. Okay, later on we'll see the bases play a very important role later on. So if we involve them in the bonding process, we cannot construct this model over here. Okay, I'd like you to spend some time to look at the notes right now to observe exactly where a typical cell will extract water molecules from. And if you want to, you can also create that model of your own on the table. Take a photo and put into your class activity as a sample for yourself. I understand you may have put in the one that you constructed. Okay, you can also include in how a cell will do it.
Fish. Right? Fish. Look, mother. Exactly.
Okay, take some time to touch up your notes. Then we'll go into the next activity. Activity 3.
My lighting is so bad.
I'm good enough.
Yes, you're touching up your notes. Do you have any questions? Okay, you'll find right that as you find this, it's actually not possible. Did you manage to put your hydrogen bonding? It's tough because of the way in which the nucleotide actually reacts. I've actually forced them all into a 2D state. But actually, each of the parts, phosphate, the sugar, and the base, right, actually they turn it a slight closer to each other. So actually on 2D, right, it's not possible.
Wait, do you take the photo in your thingy?
Yeah, your one.
You want to try
One
the
not kind, man.
use this one. Because right, there is a
What's your name?
the base, phosphate group present on top.
No, it's good.
Like this focus on
Yeah, actually, it's soft metal.
Sheep.
Sheep is the...
Okay, now I want to draw your attention to the whiteboard. Here I have a simplified model of a nucleotide. Okay, a very simplified model that I made. It still comprised largely of all the parts. Okay, phosphate group. I dropped the OH group at carbon number 3 and the base over here. It doesn't quite follow what is over here because I've orientated it in a certain way. Okay, if I wanted to, it is still largely the same. Okay, so you look at it like that. Still not quite the same. You have to flip it like that, then it looks the same. Okay, but I have to tell you right now, my model over here still has all the parts. Okay, the numbering of the carbon is still correct. It's just that mine is a mirror image and it's flipped a certain way. Okay, for convenience in a while. So, assuming that phosphate group is where carbon number 5 is attached to, OH group is where carbon number 3 is attached to. Yeah? If I were to join many nucleotides together, actually, I will always have a free end that is 5. I will always have a carbon 5 that is free. I will always have a carbon 3 that is free. Right? No matter how long or short the chain is, there will always be one end of carbon 3 that is free and one end of carbon 5 that is free. Right? No matter how long or short it is. Still following? Okay? Why I'm saying this is because there's a terminology for this. Every DNA strand has a 5' end. This apostrophe on the top, the word to describe it is prime. Okay? So 5' end. And this is the 3' end. Or if you write it out, it will be 3' end. The word prime means that it is free. Okay? It's a free end. And every DNA molecule, DNA strand will always have two ends that are free. Why is this important? Because as you construct this model over here, you'll find that one end will always be free. Actually at the very top, I actually have a free 5' end. And the opposite end below, I have a free 3' end. So you can always check yourself. Once you constructed it, the two ends need to be of different, one is 5, one is 3. Okay. So next, number 3 over here, is just to introduce to you some terminologies. Yes, question? Free end. Okay, when I say free means that actually I can form more bonds. Right? Look at this 3' end is free, right? I can actually add more, I can add more, I can add more. So that's what I mean by free. Okay? Okay, so this is a 3' end, this is a 5' end. I don't like to draw a lot on the whiteboard, so I've made and printed out DNA of my own. Okay, the next interesting thing is this. DNA as a molecule, actually there are two strands quite around each other. Right? It's not just one strand. So on the whiteboard now, I'm going to show you my DNA strand. Alright, so this is one complete strand over here. Oh, it's longer than I thought. Okay, this is one complete strand on the whiteboard. Where is the 5' end? Where's the 3' end? Same, right? Okay. So next, I'm going to put another strand of DNA. DNA is not just one strand. Okay, this is just a single DNA strand. A real DNA molecule comes in a pair. So here's my second strand.
Phosphate is connected to a 5 carbon. So then that end is not bothered to anything, so it's called 5' end.
Okay, this is my second strand.
Then
For this strand over here, where's the 5' end, where's the 3' end?
it's also 3. No, it's 3.
You can identify? So this is a skill you must pick up. You must be able to identify where's the end. So this is a 5' end. How do I tell? By the phosphate group. Okay? And the other 5' end is here. This is a 3' end. This is a 3' end.
Flipped and mirror
Okay?
image. Yeah,
So
that's why it's
this
not
is the next clue. As you are building your DNA molecule, you will find, okay, maybe I'll pause a while, that you need to take down some notes.
And then the 3' end. What's this whole phosphate? It's very long. It's 3 phosphates. Yeah. Oh, we had something called you put like
without this?
Are we supposed to label this properly?
I don't have my photo.
Oh, you want to say?
Yeah, yeah.
Yes.
Is the 5 prime and the phosphorus in the phosphate?
Yes.
Or is it the carbon?
Oh, say again.
Is it the carbon that the 5 prime end or the phosphate in the phosphorus in the phosphate group?
Are you making
Okay, the phosphate group is okay, we call the phosphate group that the 5 prime end because it is connected to carbon 5. So that's why it's the 5 prime end.
Then for example, if it's like 3, right, it's only the one all the way to the left that is the 5 prime end. Or is it the
Technically, but we don't make use of nucleotides with 3 phosphate groups. We make use of nucleotides with 1 phosphate group. Yes.
You ask Karen if you can.
Oh, are you asking like this sequence I've done over here, is it legit? Like it has to be like that? Oh, no, this is just a random sequence I made. Okay, so what pattern do you see? Remember, spot patterns. You notice any patterns
so many
in the
spots?
way they pair up? Yes. Did you see that? Notice the pattern I've done? Okay? Because in DNA, that is truly how the bases interact with each other. Okay? They pair in this way. Yes. Okay, so because even though they pair this way, the order of a single strand, is it random? I would say it's random the way I'm doing it, but in your cells, it's not random. The sequence actually holds information. Okay? How a sequence plays out, right, actually carries information for your body. Okay, it does. I feel like, I feel like for our class we are ready for this. Okay, so listen up. Actually, how the bases arrange themselves forms a message for ourselves. It is a secret code. This code is called the genetic code. Okay, we are going a few chapters ahead, but I think you are ready for it. Okay, it's actually a secret message. Okay, this is called the genetic code. And our cells, right, read the messages in triplets. For every 3 bases, it actually tells the cell a single information. You all learned in JH2 about amino acids. Yeah? Okay. And then you know proteins are made from amino acids. Okay. So actually, our DNA contains information to tell our cell how to create every single protein in your body. Okay, but how? Every triplet tells the cell to use one particular amino acid. So maybe this is amino acid A. Another triplet over here, amino acid maybe C. This one, amino acid D, and so on and so forth. So many parts of our DNA, the sequence contains a secret message that tells our cell how to link up amino acids together to form a protein. Okay, that's what we call a genetic code. Yes? You're right, okay? Aren't there some parts of your body that aren't protein, like lipids, like carbohydrates, right? Okay, so then the question is, how can our body only contain information to produce proteins? Right? Doesn't make sense when we have other biomolecules, right? But here's the thing. You know what enzymes are made up of? Yeah. Enzymes that you learn in JH2, they are made up of proteins. So actually, as long as you can produce an enzyme that can make carbohydrates, you can produce an enzyme that can make lipids, we've actually covered everything. So that's why our cell only contains information to build proteins, because we can make any enzyme we need that can make another biomolecule. Carbohydrates and lipids don't have that ability to function as an enzyme, only proteins. Other functions of proteins, other than enzymes, it can be for structural purposes. The keratin in your skin that gives you structure, that's made of proteins. The hormones that run through your body, that's also made of proteins. So actually, the cell really only needs to produce proteins, and that's why the secret genetic code of our cells contains that information. If I want to put it in layman's term, our DNA is nothing but a recipe book to produce all the proteins in our body. Yeah, really like recipe book. In a few chapters now, I will tell you who is the chef in our body. Who is the chef that creates the protein and who is the chef that reads the recipe book. Right? There's a missing link at the moment. How can a recipe book turn into the protein? Cannot. Okay? If I read a recipe book with chicken rice in it, it's not like I take the recipe book, put it in the oven, right, come out chicken rice. I need to be the chef to read it and cook the chicken rice. Okay? Sorry, yes. I see? Okay, Union? Oh, why is there no uracil? We will come to that in the next class activity. Okay? You only find uracil for RNA. Okay? So what I just shared with you here, you will encounter it again maybe 3 chapters down the road. Okay? But it's valuable information, help you appreciate biology a little bit more. Here's the wildest part. Now that you know that just by having certain sequences of DNA, you can dictate what amino acids is going to be. That means to say, technically, can we change our recipe book? Yeah? We can change our recipe book. If I want another amino acid to be over here, maybe amino acid F, all I have to do is to swap out something else. Yeah? So, I don't know if you've heard this term before, but it's called genetic engineering. Scientists now are beginning to go deep into the cells to swap out the nucleotides. Because just by swapping them out, you can swap out the amino acids that will eventually be produced. So that's what genetic engineering, the field of science, is doing. It's a very powerful but very dangerous one. Because where do we stop? Yes, we can change things up to maybe help our body, but will we cause a divide at the end of the day? Will we start tampering with the DNA to create a perfect human being, whatever perfect may be? Yeah? Who defines perfect? Yeah? So all this extra, we're going to encounter it 2 chapters down the road. For today, I'd like you to appreciate that DNA as a molecule actually has 2 strands that interact with each other. It is like a ladder. If you look at it, right? It's like a ladder. Except it's a very funny ladder. It's not just a ladder, it is a twisted ladder. On the whiteboard, I have a limitation. I cannot twist it. But in real life, can you see that the 2 strands over here are twisted? Yeah? Where are the bases over here? You see all these bases in the middle? Where are they? They are all the blue parts over here. See all the blue parts in the middle? Can you see that it is twisted? I don't know if you can see the 2 strands. Can you see the 2 strands? Yeah?
Wait, how come like the blue ones is like a hole? It still hasn't bonded with all of it.
Oh, you mean like over here, is it?
Like the blue ones, you can see there's a hole in it. Like,
Oh, okay. You can see a hole. Okay, I'm gonna, don't know if this will break. You can see holes, right, in the blue one? Yeah? Okay, if you look at the bases over here, you notice that there are rings.
yes.
Yeah? So those are the holes you are seeing. Okay, from the top view, it looks like holes. That's because the holes are these gaps over here of the rings. Really, I'm missing a bond?
Yeah, it looks like it's missing a bond. Like in the molecule, the atom itself is a hole. Like
Okay, say again. I'm trying to understand what you're saying.
the atom itself has a hole.
The atom is like over here, is it?
Yeah.
Yes, I also noticed this just. So I'm going to figure out what is missing before I pass this to you all. Hey, how you so observant? I can see this hole from here. Okay. Wait. Oh, you're looking at these.
Yeah.
Okay, you must understand that these balls that are provided over here, right? Wait, let me think. This is like, okay, all the blue ones are, you know what atom? Nitrogen, right? Notice all the bases contain nitrogen, right? So nitrogen, right? By right, nitrogen can bond to 3 things. Bonded to one carbon. Oh, so you're right. Actually, I'm missing hydrogen. Actually, I should fill up all of these with hydrogen. I am missing some parts, which I need to find before I pass it on. Okay? Thanks for spotting that. Alright, so your task is to build this. But then you notice what's holding my two strands together? The bases. Okay, so you notice that at the end of the day, one strand over here and one strand over here, they seem to be linked together. I cannot pull them apart. The question is, what's holding them together in the middle? Okay, so the thing that's holding them together in the middle is something you've actually encountered before. That's why in biology we go very stepwise to build things up. Things all tie together. The thing that's holding them together, I'm going to separate them a little bit. Are hydrogen bonds. Yeah, hydrogen bonds. Okay, hydrogen bonds. Okay, you know why we left nucleic acids as the later biomolecule? Because we build up the knowledge. The more you know, the more you'll be able to appreciate. You could have appreciated the chapter on nucleic acids if you didn't know what sugar looks like. Right? Now that you know what sugar looks like, you know that in the middle of a nucleotide is a sugar. So everything has been arranged, the sequence they are, because it helps you build your understanding. So what holds the two strands together? Lots of hydrogen bonds. We know of hydrogen bonds to be pretty weak, but if replicated across the entire DNA, it can be pretty strong. Kind of like, what's a biomolecule we've encountered that has a lot of hydrogen bonds cross-linking? Cellulose, remember? Cellulose also has the same idea. Lots of hydrogen bond holding each strand together. Instead of DNA, we only have two strands hopefully crossed together. Okay, so why is it hydrogen bond? Okay, now let's go to our next activity. Thanks for leading us so nicely. Okay, transition time. Now, how do the nitrogenous bases interact? I would like you in your groups now to focus purely on the bases. We'll now look at the structure of the bases to understand how hydrogen bonds occur. Okay? And then we'll talk about the why. Why hydrogen bonds? Why not other bonds? Okay? Why hydrogen bonds? Let's try activity 4. In reality, each pair of bases hydrogen bond to each other in different ways. I'd like you to read the sentence here. In DNA, adenine and thymine pair with each other with 2 hydrogen bonds. Guanine and cytosine pair with 3 hydrogen bonds. Your task in your group now is this. You see the big cards inside your plastic sheet? I printed out all the bases you can find, A, T, C, and G. With your prior knowledge of how hydrogen bonding works, with your prior knowledge of what kind of functional groups you need to be able to do a hydrogen bonding, I'd like you to try your groups. Can you arrange the nucleotide so that you can achieve 3 or 2 hydrogen bonds? Okay? Okay, so I'll give you around 7 minutes to complete this task.
3' is this one. Yeah.
Yeah, T1 is basically like
The hydroxyl group and the third carbon.
then what's 5'?
5' is this phosphate chain and the fifth carbon.
Some photos.
Okay, how will you orientate A, T, C, and G such that they can interact to form 3 and 2 hydrogen bonds? Yeah. Okay, so 5' and is very carbon 5 plus phosphate.
So 5' is this one all the way.
Yeah.
And then so 3' will be this C.
Right. So even as we add more nucleotides down, right, there will still always be one free 3' end, and there will still be one free 5' end at the very top.
So that means that the 5' is not
Here, sana.
Oh. In DNA, it's always one frame. Oh, they're just showing you the entire high one. Yeah, but in DNA actually we make use of the
I got a question about like the phosphate.
Yeah.
So you see the like two and the dash in front, right?
Yeah.
Does it mean that there's a hydrogen?
Yes.
Then like this here.
Yeah.
And then the there's a two minus one.
Yeah.
Okay.
Yeah. So what this means, right, is that the hydrogen is already left out.
Why?
Why is it left? Okay, so it depends on what kind of environment it's in. If it's in water, actually it can leave. You haven't learned the chapter on acids yet, but the hydrogen can leave if it's in water.
Okay, but if it's here, right, then it's supposed to be bonding, right?
Yeah,
So
correct.
here, for example, we take out the voltage, right? Yeah, we take out the voltage.
Yeah.
Then it is released to like this.
Yeah.
But
Okay, then you
it's
see,
kind of like circuit,
the orientation is just different. So actually, if this one, if I bend it sideways, right, it's the same thing.
so we can anyhow orientate it.
Yeah, the phosphate group, right, as long as it's connected to O like that, right, you can bend it upwards, bend it downwards.
So whenever we see like a dash line, we just bend to the H.
Yeah, correct.
But we can also write it as one.
Correct, yeah, you can write it as O issues. Yeah. But understand that there will always be two free O and one that's not. Yeah. Okay, this is where it connects. Okay, Wani asks, what's the asterisk? The asterisk represents where it connects to the sugar. Okay? Have you found a way to get them to interact?
Be arranged to achieve the hydrogen bonding. It's supposed to be like that.
Cannot be up down.
But there is also up down.
Because it's flip.
No, like that one. Because the
It's
two dash
supposed
is
to
supposed to be
be
where the
like
sugar
this.
connect.
Sugar don't connect.
No, just now you say the S3 means like where the sugar connect to each other. The base is here, right? The base is here. Oh yeah, we have it.
Sugar should be then like this. Then here.
Are you found a way? Okay, so G and C are three hydrogen bonds, right?
Three hydrogen. What does that mean? Three hydrogen.
Sorry?
Okay, where is T?
So you're asking is it twisted because of all the bases? Partly. So A and T needs to form how many hydrogen bonds? Two, right? Found a way? Or you look at it. But you managed to try yourself. Yeah, you're right.
See that? Yeah, this one.
Oh yes, it's C. Guys. How to bond ENT?
You say two hydrogen bonds, where got
Because
It's
of
like this.
but
Like
we
two here. But then I cannot find like the concept.
wait, this is
Jump page.
how do you make your screen go all the way back? All the way back?
No, just click your screen. Yeah. Where's expand? All the arrow.
Oh.
Yeah, but how to bond? How you all like this was like you don't even have any categorization. Where's the pig?
because the sugar is here.
to flip the it.
Yeah.
How do you flip it?
Mirror image.
Like that?
Okay?
Yeah, very
If you are done trying it yourself, then you can look at how biology does it. Okay, how do our cells do it?
Ini sama. Ini foto.
Yeah.
How do our cells do it?
Yeah, but it's missing an N. Then how do you make it?
There's this.
NH connected to
Then this one.
But where's the N?
This one here.
Theoretically possible. Yeah. So,
Let's put here.
can you show me again?
Let's put this one.
But you understand or not? You don't understand why they chose these groups. What is the basis of a hydrogen bond? What must be present in order for?
The photonic hydrogen.
Okay. Yeah. So,
The bonded to N, O, and
yeah.
Wasn't you flip this? No, this one should go on top, like. Yes.
So actually there's a slight flip.
Look.
But theory wise, right, actually you are there. Yeah, theory wise you're there. Mr. Low taught hydrogen bond already.
yeah.
Hydrogen bond, there's only NH in us. Where's the other
Hydrogen bond
H?
is here or what bond? Bond is here.
So this H2O.
Hydrogen bond you don't need H2O, you don't need water. It's just H connects to O, N or N.
Oh, the weak one, not the condensator reaction.
Yeah. Wait guys.
Isn't it like this?
Guys, this is CG.
Okay, wait. Go on table, there.
Oh yeah, there.
This one, right? Like this, right?
Yeah, hello y'all. I can do it.
So this O here, this one like this can I send it all you guys. I send them to the first.
Isn't the sugar? And you put the sugar.
Yeah, you should be correct. What? Why do you put a star in the H? What is the star?
It's sugar. And then sugar.
Wait, guys. If you're supposed to take a photo for this one, right, then why part D say repeat steps with A, C, J, E, B? Oh, the first one is E, T, G, C. Yeah, no matter what.
Yeah. It's something like this. Because in the blood type they put like remember they put like sugar is here. Metal sugar, then like if you say like the sugar over here. So it's like this.
Let me see the image. Okay, so I believe it's the same. What you chosen over here, right? Same. So this one that is different, right?
These two combined with this.
Yeah.
But it's just not drawn.
I think it's not like that.
Anyway, it's all the sugar, it's not
Yeah. Oh, top instead of the bottom.
Because they say the sugar is here.
Yeah. Hold on. Okay, so like that. And cross like that. Oh, yeah, it's like that now. So yeah, there was no need for it. There was no need for it. There's no need to flip it.
So this is
But it says DC.
Oh, C chem.
Oh, you're doing a DC, is it?
Are you signed up? Who signed up? Who signed up? Are you signed up?
Okay.
My technology
Okay, I would like to come to a close.
what happened to mine, sir?
Right.
You press to.
At the moment, we have done up to
Is there work?
Yeah.
4C. Yeah, we're down to 4C. Okay, I've asked you to try to figure out how AT and GC would bond, hydrogen bond to each other. Okay, I'm not going to do the rest of the activities. It is just, the rest of the activities just gives you a deeper appreciation. But we are a little bit pressed for time. Okay, because next week we're going to have HBL that takes up 3 periods. Okay, so we won't have the lesson. Alright, so I've crafted the remaining activities. I want to reassure you, even though we're not doing it, it's not examinable. It is just for your appreciation. It is more of a thought experiment. Okay? If you want to, you can go through these thought experiments yourself. I will provide the answers. Okay? What's the point of these thought experiments is to consider this. Why must it be A and T and C and G? Why can't it be the other way around? Right? If you really want to think about it, these are the activities you can go through to think about it. Alright? But in reality, the hydrogen bond in this way. Okay? Yeah, so at the very least, I need you to complete up to 4C to draw in the hydrogen bondings on your images. Okay, 2 dotted lines. Okay, the subsequent parts I will not be doing in class. Okay, so actually you can submit this CA already. Yeah, you can already submit this CA. The subsequent parts are all thought experiments. Okay, for you to consider. That's it. I think you can stop the